So I'm going to start today by talking to you about how genomics is already impacting on the healthcare of children with rare genetic conditions. And then I'm going to move on to talk about the promise of genomics, what genomics will do in our lifetimes to improve the healthcare of everyone in this room. So put down your Lego and listen. Okie doke, now we're in the right place. So I'm going to start by telling you a story about a patient of mine called Amina. Amina was completely well until 8 months of age when she developed a mysterious illness called recurrent apnea. Apnea is when a person stops breathing. Amina would stop breathing when her dad changed her nappy. She would stop breathing when her mother fed her. She was kept alive by the expertise of the doctors and nurses looking after her, and she had a gazillion different tests and even more medical opinions, but nobody knew why Amina repeatedly stopped breathing. We were able to include Amina in our genomic sequencing program at the Murdoch Children's Research Institute, where we took blood from Amina and sequenced her DNA. We sequenced the DNA code of all of her genes, hunting for the answer of her problems. And within 2 weeks of her blood sample hitting the lab, we had it. Amina had a DNA mistake in a gene which encodes a riboflavin transporter. Now you guys might know riboflavin, you might even take a bit of riboflavin, it's vitamin B2. Amina's condition, exquisitely rare, in some cases responds to high-dose vitamin B2 therapy. We started this treatment, and within 4 weeks of starting the treatment, Amina's apneas had gone away, and she is now out of intensive care and home with her family. My clinic is full of undiagnosed kids with suspected genetic conditions whose health and development suffers because we have not, up until now, had the tools we needed to make a diagnosis in them. Genomics has the power to change that. We've sequenced more than 100 kids' genomes looking for the answers for their genetic problem, and we get an answer, a diagnosis, in more than half. When we compare that to the usual tests that we would do before we had access to genomics, which was things like a brain MRI under general anaesthetic, sampling spinal fluid, running metabolic tests on urine and blood, these tests being painful and expensive and invasive and time-consuming, genomics gives us 5 times as many diagnoses. 5 times as many diagnoses at a quarter of the cost per diagnosis. What our research tells us is we need genomics for kids in our clinic, and we need it now. But what actually is genomic sequencing? Genomic sequencing is the spelling out of a person's DNA code. Spelling out the code of individual genes, looking for changes or mistakes. Each gene codes for a particular protein in the body, and proteins are the building blocks of our body. If a person has a mistake or a change in a given gene, that might make a faulty protein or no protein at all and lead to disease. Now, we've been able to spell out individual genes for decades. We could go one at a time and sequence one individual gene at a time quite well. The breakthrough in technology has been that we can now do them all at once, all 20,000 genes, all at once. And we can do it fast. We can sequence a person's genome, analyse the results, and return the results to a family in 50 hours. Sort of what you might do on your average weekend. So it should be pretty simple, right? We should be able to see a child with a problem, grab a blood sample, sequence their DNA, find the answer, return it to the family, head off for a coffee. Well, there's still a bit of work for us to do. We still have a lot to learn about telling the difference between harmful DNA mistakes and harmless DNA variation. Computing has massively sped up how quickly we can sequence and how much DNA we can sequence, but a lot of the analysis is still done by humans, and that makes it slow and pretty fallible. So in our hands at the moment in our clinic, the test takes 4 to 6 months and costs us $2,000 per person. So what do we get for those $2,000? Well, we get the answers that I told you about before, the answers that families are looking for. What is wrong with my child? What does the future hold for my child? And what are the chances of this condition happening again if I have more kids in the future? One thing I really didn't dream of when I started this project was that we get more than just answers to those questions. In a third of the diagnosed kids, we get changes to their treatment. So that might be a dramatic change, like in the case of Amina, or it might be a more subtle change. We might learn that because of the genomic diagnosis, we should avoid a particular medication which is contraindicated in those kids. Or we might learn that because of the genomic diagnosis, we should keep this child's kidneys under surveillance for particular complications that can occur in the future. I was blown away by how often the genomic diagnosis led to better care for the child. And we need these answers and we need these changes in care, because if you add up all these exquisitely rare genetic conditions and other rare conditions in our community, they affect 1 in 12 individuals. 1 in 12 people have a rare disease. Many of these diseases affect children, many of them are genetic, and many of them are undiagnosed. So people with rare disease, and particularly children, need genomics first and foremost. But they won't be the only ones who benefit from genomics in the future. So looking to the future, it's incredible where genomics could take us. The fact is, and I'm sorry to tell you this, but you're all genetically not quite right. Knowing how can help our medical care. Let's take medications, for example. The way we use medications in our society at the moment is pretty much you go and see a doctor, they diagnose an illness and they give you a tablet. They give you the same tablet pretty much no matter who you are, where you're from, what your ethnicity is, etc. It's a one-size-fits-all approach. But the clearinghouse in our body for medications is the liver. And the way that our livers work varies enormously from one person to another. Some of you will have a Usain Bolt type liver, a bit of a sprinter liver. You might be the ones who can knock back a few beers and not really notice it very much. A sprinter liver will metabolise drugs really fast. And a person with a sprinter liver is going to need a lot more of a given medication to get a good therapeutic effect. If you compare that to, say, a more sluggish liver, a liver that metabolises quite slowly, a sort of tortoise-style liver. A tortoise liver, or a person with a tortoise liver, is going to metabolise drugs slowly and be very sensitive to particular types of drugs and get side effects at really low doses. The secret behind who has a tortoise liver and who has a sprinter liver is, of course, in your DNA. And doctors can't tell at the moment who has which type of liver, so we treat everyone as if they have a slow, sluggish liver. It's a bit like if you imagine all of us trying to fit into the same sized pair of bathers. I don't know about you, but a pair of budgie smugglers are not going to meet my anatomical needs now or in the future. If we can understand our DNA and understand how our body metabolises drugs, then we can tailor the choice of drug and how it's used to that individual's type of metabolism. Genomics is also going to change how we treat cancer. Cancer is going to affect 1 in 3 individuals in this room in our lifetime. Cancers happen when a group of cells break off from the herd and start to multiply out of control. Normally inside our cells, we have genes encoding proteins that act like a kind of brake on the cell. They stop the cells from multiplying like crazy. But if you acquire mistakes in those genes, the brakes fail, the cells start to multiply, and that's what makes a cancer. Traditional ways of treating cancer used drugs that killed rapidly dividing cells. So that was good because cancers rapidly divide, and so some of the cancer cells would be killed. But those drugs also killed healthy rapidly dividing cells like skin cells and blood cells and gut cells. And so those drugs have a really serious side effect profile, which sometimes was worse than the cancer itself. Using genomic technology, we can target the specific DNA mistakes that are driving that tumour and develop medications which are already in clinical use for some cancers that don't randomly kill cells in the body. They target the cells with the cancer genomic signature of the tumour. So we can maximise the therapeutic effect of the drug and minimise the side effects for the person. Genomics is a game changer in healthcare, and all of us in this room are going to benefit from the way that genomics will impact on how we are diagnosed and how we are treated when we're sick. There's an amazing, untapped knowledge base inside each and every one of us in our DNA, and we're only just beginning to realise its potential. Thank you.